Hi, my name's Amy, and I'm going to explain randomized controlled trial designs, also known as RCTs. RCTs are experiments or studies that aim to reduce bias when testing the effectiveness of new treatments. In clinical research, RCTs are the best way to study the safety and efficacy of new treatments. They are the gold standard of research and considered the most rigorous and robust research method to determine if a cause-effect relationship exists between an intervention and an outcome. The quality of an RCT depends on a variety of factors, but most importantly, reducing the risk of bias. Bias can under or overestimate the true intervention's effect. Choosing the appropriate RCT design is crucial to produce data that can be translated into clinical practice. The Consolidated Standards of Reporting Trials, or CONSORT, is a minimum set of recommendations for reporting RCTs. It's basically a template or guideline for authors to follow and report findings. The CONSORT statement is a 25-item checklist and a flow diagram. The checklist focuses on reporting how the trial was designed, analyzed, and interpreted, while the flow diagram displays the progress of all participants throughout the trial. A well-defined protocol should be written and reviewed by an independent ethics committee before starting the trial, and the trial should be registered in a public trial database. The initial design of an RCT must start with a hypothesis and a clearly defined research question. A good research question should use the PICO format. As an example, let's say researchers have come up with an incredible drug that will stop or decrease the urge to smoke. Let's call this butt out. An example of a research question might look something similar to this. Once the research question has been established, the next step is to identify the population the researcher is interested in studying, known as the target population. If we look at our fictitious drug, our target population would be smokers. Once the target population has been identified, a suitable sample must be recruited for the study. Inclusion and exclusion criteria must be defined to determine who is eligible and not eligible for the study. This will then become the study sample. If we look at butt out again, we know the target population is smokers, but what criteria will we use to determine who goes in our study? This then becomes our sample. After a suitable sample has been determined, an appropriate sample size must be determined. The sample size must be large enough to allow the study to detect a clinically important difference between the groups being compared. Poor sampling, or sample sizes that are too small, can undermine results and reduce the validity of the study. Selection bias can occur when sample selection does not accurately reflect the target population. In an RCT, as the name suggests, subjects are randomly allocated to two or more groups. One group, the experimental group, is given the intervention being studied, while the other group, or groups, called the control group, is given an alternative treatment such as a placebo or a conventional treatment. The groups are followed under the trial design to assess how effective the experimental treatment was compared to the control groups. Blinding is another very important part of an RCT design. When possible, it is essential to blind both the participants of the study as well as the researchers involved. This is called a double-blind RCT. If double blinding is not possible, this must be listed as a limitation of the study. Blinding is important for ensuring internal validity and reducing observer bias, performance bias, and detection bias. Another aspect of blinding is ensuring both therapies look identical, such as each pill looking the same, as well as all participants receiving each therapy in the same way so that no bias can be introduced. Allocation concealment is another important part of blinding. This means that the person randomizing the patient does not know what the next treatment allocation will be. This is important as it prevents selection bias affecting which patients are given which treatment. A primary endpoint is a key factor in an RCT design and should be defined before the study starts to ensure objective reporting of the findings. The primary endpoint is basically the main result that is measured at the end of a study to see if a given treatment worked. If this is not defined until after the data is reviewed, researchers could choose the data that confirms their bias. Both significant and non-significant results need to be objectively reported, as well as identifying the magnitude of errors that are acceptable. The consort statement should be followed, and any potential conflict of interests and funding should be listed. The beauty of a well-planned and designed RCT is that bias and errors can effectively be reduced or designed out. While I don't have time to discuss all aspects of an RCT design, I've touched on the major features. Thank you for watching. I hope you've enjoyed this presentation.